Okay, while they're getting set up, I'd like to introduce Dr. Serge McGraw. Dr. McGraw's main research focus is on understanding DNA methylation issues like those in TBRS. His research laboratory aims to understand how these issues may be involved in developmental disorders. This important research helps to understand the nature of disorders like TBRS and brain development. The aim is eventually to design targeted treatments for brain disorders. So please help me welcome Dr. McGraw.
First of all, thanks again for the invitation. And like I'll say, like everyone, it's quite a meeting that's special for us because we never get the chance to, for me anyway, to interact with families and patients. So I really appreciate it. So today my talk is going to be mainly on the different models that we have in the lab. I'll give it a little bit of data just to give you an overview of it, but it's more to explain how we create these models and how are we using them at the moment. So you saw a different version of this. I'm going to give another way to explain it, just to put you in the mindset of why and how important is DNMT3A and methylation. So DNA is the genetic information inside of our cells. It helps us make who we are. DNA will act like codes in a video game or blueprints for a house. Here you have a cell, you have the different genes on the DNA, and then these genes will create protein. However, depending on the cell types, depending on the time of development, genes will be expressed or not. And this is mainly regulated by chemical switches, which are called epigenetic marks, and they'll turn gene on or off, but it's also like a regulator. So sometimes genes might be overexpressed or like a little bit less expressed. So this is all regulated by epigenetics. And today I'm mainly going to focus on DNA methylation. So if we go back to the first cell of the body, so the zygote. So the zygote is a oocyte that has been fertilized by the sperm. These cells will divide into little embryos, create a blastocyst that will implant, and then these cells will generate every single cell that we have in the body. So if we look across these cells, they all have the exact same genome, so the same DNA. So for about 200 cell types in the body. So the only difference between each of these cells is the epigenetic marks. So the little chemical marks that are present in every cell type that will, drive these cells to be who they are. For example, if you take these cells, we call them embryonic stem cells. They will derive, you can be deriving all kinds of cell types if you push them towards these cell types. So here, when you have differentiation of a cell, for example, this is a skin cell, so the little chemical marks will be put together and they will express genes that are needed for skin cells. So epigenetics is critical for development. So for normal development, you need these specific set of marks or specific little chemical tags. If you perturb this, this can lead to abnormal development or diseases, or cancer or whatever. So how it works is throughout time and throughout, differentiation from a small embryo to a somatic tissue at the end, you have genes that needs to be expressed and genes that need to be repressed. For example, if we take this gene, let's say that it needs to be expressed throughout development, and this will lead to normal brain development, normal heart development. If you have an epigenetic dysregulation, so an error in DNA methylation, for example, and it shuts down this gene at not the proper time point, or it shuts down the gene where it has to be expressed, then this can lead to altered development. And sometimes that's why, you can have a specific gene that is abnormally regulated, will cause defects in the brain, but can also cause defects in the heart, because at the earlier time point, when cells are being derived, it also depends, where the epigenetic mark was supposed to be, or if there was an error into it. So this is, this is why it's so complicated to study epigenetics. So like we mentioned, DNA methylation is installed by DNA methyltransferases. DNMT3A and 3B, they kind of act similarly. They have specific regions for each of them to put methylation, but they still have the same kind of mechanism. So they will install methylation where there's no methylation. So their job is to put methylation down, you know, they're putting the first layers of bricks. Then when cells are dividing, so you're dividing your DNA, what happens is then you have DNMT1 that will maintain this profile. So if DNMT3A is not working properly and is not installing methylation, so DNMT1 cannot maintain these profiles. And if DNMT3A is installing methylation in an abnormal way, then DNMT1 doesn't care. It will just go there and maintain these erroneous patterns. So if we go back to DNMT3A, we saw these cartoons across our presentation today, but I just want to highlight that DNMT3A works in a quadruplex. So there's 4 proteins. There's 2 DNMT3A proteins that we see here. So these 2 here, or the green protein that we see here, the green structure. So there's 2 DNMT3A proteins. And then there's 2 DNMT3L. So 3L, they're just like a helper. They come here and they will help 3A do their function. So depending where your variant or your mutation is, you know, you will have either a protein like Dr. Gabel mentioned earlier on that will be degraded, you know, they'll be crushed like the cars that were crushed. And if you have other mutations, that might impact the stability of this complex. So for example, we have one of the mutations we're working on is right at the boundary of 3A and 3L. So what happens is we think that this, you know, this contact between 3A and 3L is being disrupted and the complex is less stable, so not able to do its function. So these are the 2 mutations that we're working on. So the one we call 688, it only changes 1 little letter in the code, so the protein is still there, just that it's not functioning properly. That's the mutation at the boundary that I was mentioning. The 2AT mutation, the one that's here, it causes the protein to be degraded. So what happens in the cells, so because patients are heterozygous, you'll have complexes that will be formed with normal DNMT3A, and then for the ones that the protein is completely degraded, then you'll have normal complexes, but just half of them, and then for the 688, you'll have DNMT3A that's non-functional. So if we look just at the levels of these proteins, this is exactly what we're seeing. So this is control cells. These are the one with the 2AT, so half of the protein. So this one has normal levels, but you know, only half of it is working properly. So when we study DNA methylation and we study brain development, we actually need to look at the cells of the brain. So when you do genotyping, for example, and you want, like, for your kids that have been, you know, diagnosed, probably they draw blood, or some you may have been vocal swabs. So when you look at the DNA, that, you know, it doesn't matter. Like I mentioned earlier, all the cells are the same. They all have the same DNA code. However, when you look at DNA methylation, you actually need to look at the specific cell types. So that's why in the lab, we focus on brain, we focus on brain across development, and we're looking at this on and off switch, or that, you know, that locking mechanism. So we use stem cells to look at brain development. So this is a stem cell. A stem cell is a cell that has the potential to be derived in all kinds of cell types. So, but also stem cells, they can also renew. So you have a stem cell, it just copies itself, like a photocopying machine, you know, copies itself, everything is identical, and then when they're ready, they can be differentiated into specialized cell types, which, you know, can be heart cells or brain cells or all kinds of different cells. Where do we find these stem cells? Well, you can find them in embryos. So these are first stages embryo. They go into a blastocyst just before it implants. You can also find stem cells in different adult tissues. But if you look at embryonic stem cells, you can use embryos. You get these cells out of the embryo. You put them in culture, and then you could grow them. You can, you know, modify some genes and then differentiate them into specific tissues, and then you can look at the mechanism and the function of these cells, these genes, for example. So we call them pluripotent stem cells because they can be derived or differentiated in any kind of cells. And the way that we do it is just by changing the media. So we put different conditions or different factors in that cell growing media, and then we can grow them into skin cells, neurons, and like liver cells. So this is, one of the way to do it. So using cells that are derived from really early embryos. But this, it's not really possible for what we're doing for TBRS. So what we're doing in the lab is we're taking somatic cells. So we took blood cells from patients, and what we do is we force the expression of certain genes in these cells. So by forcing the expression of these genes, we're reprogramming these cells, and then the cells that were skin or blood cells are turning into stem cells. And these stem cells then, again, you can put them in culture and then differentiate them into different cell types. So what we're doing in the lab, we use these iPS cells, put them into neuronal stem cell. So once they took that path of neuronal stem cells, they will be able to be differentiated into neurons, interneurons, or different types of cells. And all of these different types of cells have different function in the brain. So if we believe that, for example, there's issues in terms of connectivity in the brain, so we can derive them into interneurons, and then we can look at aspects related to brain connectivity. So we do this for both of our cell types, the 2AT variant and the 688. So what we're doing with this is we're actually looking at how the genes are affected, but not only the genes. So we're looking at the mechanism. So we know that the main regulator is the NMT3A. That's the kingpin at the top. But what's trickling afterwards? In this, you need, again, to look at specific cell types. So this is how we're using our iPS cells. And then we're trying to define, is there a specific cell type that is more affected? So we're using, high confocal microscopy and looking at specific proteins that are, abnormally expressed. And the end goal of all of this is, like many of us, have mentioned, is once we have identified that specific mechanism, then we can go and test. So if we put back the NMT3A, can we rescue some of these phenotypes? If we treat it with a small approved molecule, if we treat it with a specific drug, so how can we help these cells be more normal? And again, do we have to do it in the progenitor cells, or can we do it at a different, at a later time point? So this is all questions that, we're addressing. So if we go back to our iPS cell model, so there are cells that are stem cells, for example. So here we have an example of a stem cell colony in a dish, and then Karine, that's in the lab, she's pushing these cells into neuronal progenitors, and then we're specifically looking at forebrain cells, so this the cortex. And when we do this, we already, in the iPS cells, so the cells that are, the ones that are pluripotent, we already see that there's errors in the genes, but also in the DNA methylation. But these errors are, there's errors there, but they're not that striking. But when we start pushing the cells towards differentiation, that's where we see that you also have the errors that were original from the iPS cells, but you also have new errors that are being created in terms of genes that are abnormally expressed and also the methylation that's being laid. So for example here, if we look at the DNA methylation, so we have here PBMCs are the blood cells. So iPS cells are the ones that have been reprogrammed, and then the neuronal progenitor cells. So if we look only in the blood cells from patients and compare them to control cells, we already see that there's errors in methylation. When we reprogram the cells, as you can see here, there's errors that are overlapping the blood cells. However, some of the errors that we saw in the blood cells are not present in the iPS. If we push the iPS towards neuronal progenitors, again, we have errors that are maintained, and we also have errors that are present in the PBMCs, the stem cells, and the NPC, but we have all these new errors that are here. So again, the further along the pathway of differentiation, 3A, because every time you differentiate your cells, 3A is adding new methylation, to repress specific genes, But because the gene is abnormally functioning, then you're dragging the old errors, but also you're creating new ones. And then when you look at the genes that are associated with these errors, they're all related to brain development or brain function type genes. So this was for methylation. So what happens just for the gene expression? Because methylation, it's a signature, it's a pattern. But what's important is also the genes. So again, we have, so there's about 30,000 genes in the genome. About 200 are affected in about 300 are affected in the iPS cells. Again, when we push them into NPCs, we see a lot more. When we push them into neurons, we see, again, a lot more. And there's not a lot of genes that are maintained throughout. So there's always, depending when genes need to be expressed. So if we divide our data sets into genes that are really key for all these specific neuronal cell types. So, so these are just genes that are regulating all kinds of neurons, but then you have glutamergic and then GABAergic. So these are the ones that are super important for the connectivity in your brain. So on this side here, on the left side, it's the control cells. On the right side, the three rows that are there are for our mutant cells. So as you can see, all of these genes that are key in neuronal differentiation or neuronal specification are being impacted. So now we're using this model and we're isolating specific cell types and seeing, again, which are the common pathways, and can we play on these pathways by either regulating the NFT3A or other genes that are important to drive the expression of these factors. So another model that we just introduced in the lab, again, Karine is working with a couple of students, with collaborators of ours. We're taking these patient-derived iPS cells, and so cells usually grow flat in culture, like you see here. We call them 2D, so they're a sheet of cells that are growing. So we're, again, treating these cells with different media, and now we're able to grow these cells in little round spheres, which are tiny, like 1 millimeter, and they recreate certain aspects that we can find in the brain. So by doing so, we have these little structures, and we're able to see, which structure is impacted by the variant. But also, you have to remember that these little brain structures are not as complex as the real brain. So if you have genes that are abnormally expressed, the impact on the growth or the specification of the structures is going to be amplified in these models. So when we take control cells compared to our two variants in the NMT3A, we can see that our 2AT and the RR8, they grow larger in terms of sizes. And then when we cut, we do sections, and then we look at specific structures or specific function. Here, this is a control cell again. What we usually see is these lumens. The lumens are like little tunnels. So you have these little tunnels, and these tunnels are layered with these cells that are highlighted in red, and these cells they proliferate, and they need to be there so that, to produce the neurons. So the green cells that we see are different kind of neurons. So when we look in the 2AT variant, we can see that we lose these little lumens. So these little tunnels are not there anymore. We still see the red cells, which are proliferating, and we still see the neuron, but the structure is being altered. When we look at the other variant, here we lose these defined lumens, and then we end up with big cavities, and these progenitor cells that usually are all around have been differentiating in other cell types, and what happens is, again, we lose that structure, and if you look here, we don't see a lot of green, so there's not a lot of neuron type cells here. So again, we're using this, we're trying to put back the NMT3A and test if with different other genes that we think are targets, and see, can we improve it, and when do we have to improve it? Can we improve it once the structures are already established, or do we need to do that really earlier on? So this is an example of also something we're doing. So here, this is the variant that I was mentioning earlier. So we have half of the protein, but all the DNMT3A is all normal. So we're tagging the DNMT3A protein with a fluorescent tag, and then we're using drug screen. And the goal at the end is to see, can we either boost the 3A expression to have it to the normal level, or can we impede its degradation, meaning that there's still going to be a bit less, but they're going to be there a bit longer. Again, when do we have to do this? Can we do it? Do we need to do this in really early embryonic cells, or can we do it later on? And finally, with the help of the TBRS community and the rare disease model in Canada, what we did is we generated another DNMT3A mouse model. So our model is based on the 688 mutation. So in the mouse, it's called a 684. So we just got these mice early spring, and again, we already see the phenotype, that obesity phenotype, and like Harrison is going to probably showing you after my presentation, we are starting to see all these little behavioral abnormalities in the mouse model. So again, we're going to be able to use the mouse model, go for the mechanism, and the thing is, and what's nice, because we're probably sure that our mutation is going to be different from the other variants, so we'll be able to compare notes and compare our models and see, what's similar and what's different. So the things that are similar are probably what's driving TBRS. Again, doing high resolution imaging across development, in young adults or in adults, and as well, what we found in our cell model, then we're going to bring it here and try to use it in terms of drug screen. So our project is mainly to identify the specific pathways that are altered by the dysfunction of 3A. Compare with other TBRS models that are out there, compare also with other disorders that are related, like for example the EZH2 that we saw earlier. Which cell type? So is there one cell type that is mainly affected? So if we can pinpoint 1 or 2 or 3 cell types that are the main drivers of some deficits, then can we target these cells? So the perspective is really to, use as well the patient registry. So right now what we're doing, we're looking at the registry. So for example, we saw things that we didn't know before, like we didn't know in the lab, like related to muscle. So now we're using our mouse model, and then we're doing muscle studies. And there's things about the heart that we can do, and there's things about the scoliosis, and all kinds of little things like that that we're pulling out of the registry and using that to drive the questions behind our models. So this is my team. The TBRS DNMT3 work is mainly driven by Karine, that's sitting here, Diego, Anthony, and Josiane. So it's a big group effort from my team, and again, thanks for the support, and thanks, for the invitation. And I'll be here all day, so if you have questions, just feel free. And if I haven't met you, some of you, please just come by and shake my hand. I'll be honored. Thank you.
Okay, I'd like to introduce the last speaker in this block. Dr. Harrison Gabel received his bachelor's in molecular biology from Princeton and his PhD in genetics from Harvard. He's currently an associate professor in the Department of Neuroscience at Washington University, St. Louis. Dr. Gabel's research group has a broad goal of understanding how mutations can lead to autism and related neurodevelopmental disorders. Dr. Gabel's research group has developed multiple mouse models of TBRS. In ongoing studies, his group is investigating potential therapeutic approaches for TBRS. So please welcome Dr. Gabel.
Great. Well, thanks so much. I realize, thank you. I realize I'm the last of maybe several very dense science talks, so I hope you can bear with me and I can add something to the conversation. One apology. As scientists, we try to put together talks that hold together on their own, but we don't have time to necessarily sort of compare notes of what are you going to do, introduce, what are you going to introduce. So there's going to be some repetition here. Hopefully that's helpful for solidifying some of the real basics about how DNMT3A works and how we're trying to understand the disorder. I'm probably just going to repeat a little just to keep it coherent, but my apologies for that. So I'm Harrison Gabel. Thank you very much for the introduction, and thanks so much for the opportunity to be here. This is a unique opportunity as a researcher in terms of inspiration for the work we do. The commitment that you guys make to come here to be a community that all the volunteers make sort of is such a kick in the butt for us to go work harder and do more for you guys. So it's a really transformative experience as a scientist to be at this meeting. I brought my brand new student Kim here so she could witness that, and so thank you so much, and I really hope that we can continue to contribute to understanding and push for therapies. So very quick introduction to my lab. The lab was started in 2015 at Washington University, and serendipitously for my work in TBRS, we really were trying to understand the basic biology of DNA methylation in the brain. That's what DNMT3A does. We were working really to try to understand basic biology, neurodevelopment, how genes get turned on and off. We did have a focus on Rett syndrome, which is another neurodevelopmental disorder caused by disruption of a gene that reads out DNA methylation. So when it emerged that TBRS was caused by DNMT3A disruption, we felt like we were in a really good position to contribute to understanding the disorder and dove into understanding DNMT3A variants and how they impact cells and how the disorder proceeds. I'm going to be talking about work done in the lab today. Really the powerhouse behind this has been Diana Beard, who has been a graduate student in my lab for the last several years. And Kim, who's here, is learning about the disorder and is going to be picking up the torch and continuing it on. So those are the folks really driving the research. I'll tell you sort of an overview of some of what we found using our mouse models, and this is really complementary to what Dr. Gabel has talked about and Dr. McGraw and others. And then a little bit of a preview of what we're doing with some of the cells, similar to what Dr. McGraw was talking about, that have been donated by the TBRS community, so your contributions at work. So we'll dive into the mouse models. Very briefly, we're going to try to use mouse models to understand what happens when the DNMT3A gene gets disrupted and sort of at a molecular level first, and then how does that impact the organism. Just wanted to make sure we're all on the same page here. This is your review from earlier today. So the DNMT3A gene is in the genome, in the DNA. It then sort of produces a protein that has a job in the cell, a little molecular machine inside cells. And so when you're seeing a variant in your loved one's DNA that affects the DNMT3A genes, the change in the DNA, but that impacts the function of the enzyme itself, the protein that is produced, so it can't do its job. And as we've heard in sort of more impressive detail that I'm showing here, what DNMT3A is supposed to do in cells is put in this chemical mark, and every cell in the body of someone that does not have TBRS has 2 copies of the DNMT3A, and that produces a certain amount of the DNMT3A protein that is putting in the methyl mark in the cells on the DNA. And someone with TBRS, they're missing half of that. They have 1 good copy and 1 copy that's no longer functioning as well. And so we expect that there's going to be too little DNA methylation in the cells, and this is going to impact the overall function of the organism in the case of a mouse and, of course, individuals with TBRS. So we're using mice to study the disorder. I just wanted to very briefly highlight from a sort of 10,000-foot view why mice are so useful. Mice have all the same organ systems we have and pretty much the same genes we have. So they're obviously a good model for how all the genes work, and they have this very long track record of being able to study the causes and look for treatments for numerous diseases. And so, of course, mice, we can create lots of mice and study them in the laboratory quickly, and that pushes the research ahead rapidly, as we hope we're doing for you in our work. So the simplest thing to do is to create a mouse model where we create pretty much the same type of disruption of the gene that is occurring in TBRS. 1 copy is disrupted, and that's what we've all been doing, and you've been hearing about from these talks. I did want to highlight very briefly what what these models can give you. We've heard individual sort of explanations of examples, but at a higher level, once we establish mouse models that have some symptoms, some phenotypes that look like the disorder, we can do any number of things at any number of levels. So the first thing we can do is ask what organ systems are affected and what's the sort of cellular basis by which that's occurring. So if there is long bone overgrowth, we can, that creates taller height. we can go in and look at the cells and the bones and ask exactly what's going on dozens of mice, so we'll be sure about what's going on. Other organ systems such as the heart or the blood, again, we have this reproducible system to be able to go in and really find out what cells are affected. This gives us potentially a chance to look at the molecular underpinnings, really at the level of inside the DNA in the cells what's going on, and this can give us ideas for rational therapeutics, how can we fix that. And so once we have those ideas, we can take them, we can sort of generate the therapeutics we want to try, or go find them and bring them back and test those therapies in the mice. So sorry for that repetition, but hopefully it's helpful to be thinking about sort of why are we all making mice all the time. So what have we done? Well, a number of years ago, we started our studies by making the first and sort of simplest mouse model that you might make when studying a disorder, like TBRS, where we think it's the loss of the function of the gene in some cases that's causing the disorder. This is to basically do what we call a knockout, so delete a portion of the gene so that it is basically non-functional, and study what happens. And so we published this paper 3 years ago now, where we made this heterozygous knockout mouse, which does replicate some individuals with TBRS. We've talked about how many have this single sort of letter change missense variants, but there are individuals where the whole gene is deleted, and this would be sort of replicating that effect. So was this a good model? We think so, and I think that we've continued with the other mouse models to see that this is a disorder that can be studied in mice very effectively using these models. The mice have overgrowth, and we've heard about them having more increased body weight and obesity. We've done a number of behavioral studies. I'm not going to get into the details of the mouse behavioral studies. Mice are not little humans, and they cannot replicate all the behavioral challenges in TBRS, but we can measure changes in animal behavior in various domains of function, such as motor function, anxiety-like behaviors, and actually social behaviors in mice that can report on the overall cognitive function and nervous system function in the animals. And we see changes in these mice that suggest we'll be able to study this and potentially study our ability to improve this in the mice going forward. And then at the molecular level, which is a lot of what my lab does, we looked at this DNA methylation, particularly in the brain, and saw pretty big changes in the DNA methylation, exactly as we would predict that there was loss of DNA methylation. So I think we have a pretty good model here, but we didn't want to stop there because of this issue that there's so many different variants in the disorder. As Dr. Tatton-Brown mentioned, it's mostly missense variants, right? Changes in one letter. And so we don't quite know why it is that way. A lot of disorders are not that way. But if we're going to have a sort of a complete compendium of useful reagents, we think that we need to have mice that have this type of variant. And so Dr. McGraw has a great model that he's developing. Dr. Gabel is developing models. And we've developed, partially in collaboration with Timothy Lay at Washington University and partially on our own, 2 more models that we're studying. And that's this R882H variant that Dr. Tatton-Brown mentioned is so common in the disorder, and then a 904L variant. We have reason to believe going into these studies that this actually at a molecular level, in terms of what it does to the gene, might have a bigger effect and maybe a bigger impact on the mouse than this variant, which has, in our molecular studies, was slightly less deleterious to the function of the protein. So we thought this was, and it's turning out to be, a way to look at the potential for the variability and the severity of the disorder to be coming from the genetics of the disorder. Dr. Tatton-Brown talked about genotype-phenotype correlations and trying to understand this from patient data, but it's very hard to study because of the normal variability in our population. Mice are identical genetically except for this variant, so we can really isolate the effect of the variant between these 2 different types of mice. So this is just restating what I guess I went through just now, trying to say what can we use these mice for. We can look for the common effects across all of our mouse models. These would be sort of core mouse TBRS-like phenotypes. We can look at the variability between the mice, and we can really be modeling the various variants that individuals have. To summarize very briefly, we looked at overgrowth in these models, and we see that they all have overgrowth. So once again, this is turning out to be sort of a system. The mice are really allowing us to look at key features of the disorder. We did an array of behavioral testing, like we did with the knockout mouse, the heterozygous knockout mouse, right here in our new studies. What's interesting is we do see a differential severity of behavioral effects, starting with the 900L, which is the mouse version of the 904 allele that I mentioned, or the variant that I mentioned, and then with the 878, which is the mouse version of 882, at the other end of the spectrum with many more effects across different types of behavior that we studied. So we think this does represent some of the genetics causing a differential severity of presentation and effects on the nervous system in the mice. And now we can really study this, in a system where we have control of all the rest of the genetics, everything else, and understand, what might be driving the more severe effects in this one mouse model compared to this other one. Speaking of the molecular effects, now that we have these mice, we can go and look at DNA methylation happening in the cells. So just to highlight, the idea is that DNMT3A has a function in the developing brain and in other organ systems, in neurons as they're born, or within the functioning brain in adulthood, where inside the neurons, the brain cells, there's the DNMT3A enzyme inside the nucleus. It's putting in this DNA methylation and it's doing its job. But when we disrupt DNMT3A, this leads to a change in methylation. So we went and measured that in the brains of these mice that we had generated and asked what the effects are. And we see a correlation between how severe the effects are on this molecular mark inside the cells, the brain cells of the mice, and how strong their behavioral effects were, where these 878 mice have a bigger effect on certain types of DNA methylation, in particular special forms of DNA methylation that happen in the brain. So we think we have this correlate of some of the differential severity in phenotypes and symptoms in the mice with the molecular changes, and we're going to continue to study this. Finally, I just wanted to add that we're able to start taking these molecular signatures we have and comparing them to other models of neurodevelopmental disorders that we think might be connected to TBRS. So, for example, Soto syndrome has been mentioned a little bit yesterday and today. Soto syndrome is another overgrowth and intellectual disability disorder characterized by Dr. Tatton Brown. It's caused by mutation of a gene known as NSD1. And then there's another disorder known as Rett syndrome that's caused by mutation of MECP2. We as molecular biologists recognize from some studies that have been done outside of the brain and outside of these disorders that they actually have functions inside the nucleus that help DNMT3A do its job. So we started looking at whether or not the changes in the brains of those mouse models look like the changes we see in the DNMT3A variant models, and we in fact see overlap. So we think that there's really a convergence at a molecular level of some of these disorders. And Soto syndrome and TBRS in particular have quite similar clinical presentations. And we think part of that is because there's such a close connection at a molecular level. So this gives us sort of convergent pathways that we could think of for common therapeutics potentially in the long run if we can better understand them. So this is a big list of everything we characterize in these mice, and I pretty much have covered it. I won't go over it in a lot more detail, other than to say we now have 3 very good models and that give us both the core effects that we're seeing across all the models and variability, just like what you see in individuals affected by TBRS. And we can study this at the organismal level and at the molecular level. I'm big on summaries, so this is pretty much saying the same thing. In the interest of time, I won't reiterate it. I did want to talk about where we might be going, or where we are going, might be going. There's lots of things we might do. But, we've been characterizing the mice, and we saw on the research roadmap trying to understand the function of the gene, get models and understand sort of the organismal impact. I think we've done a really good job of getting there, and we have lots of labs working on them. Now I think it's really important that we stay sort of eye on the prize and think about how we can use these mice to start exploring what therapeutics might look like. And this is a process that can be sort of a winding process where we have to try lots of things, but we have the tools, so we should really start trying. And really what we want to be doing is trying to devise what a therapy might look like, testing it on the mice and seeing if the mice improve, both in their behavior, in their physiology, and at the molecular level, because we have such good insight into what the DNMT3A gene is doing in the mice. And then we can start testing things like, when would you need to give this treatment in order for the mice to improve, something that's really, really important in a developmental disorder like TBRS. I did want to highlight one experiment that we're doing that we think is important, looking pretty far down the road, but is becoming a really key experiment to do in various neurodevelopmental disorders, where we're trying to establish if something like a gene therapy is a viable option for the disorder. So a developmental disorder, it's possible that some of the changes, as Dr. McGraw was mentioning, some of the changes that happen in early development due to the lack of the gene are not something that you can correct when you put the gene back later, right? You'd have to look for other therapies because development has already happened. However, there are some disorders that were originally recognized as neurodevelopmental disorders, where if you put the gene back, you actually have quite a big impact on the overall symptoms and disorder, and it's quite promising. Gene therapy is not ready for neurodevelopmental disorders yet, but it's actually something that the biopharmaceutical industry is starting to look into. So it's not this year, but in the coming several years, it is something that if you could demonstrate that a disorder is a good candidate for this, you could attract interest in doing these types of therapies. So we wanted to do an experiment that tested the idea of, if we could do perfect gene therapy, how much improvement could we get in mice? And if that looks really good, then we would really continue on. If that doesn't do what we want it to, then we think about other therapies. So that's what Kim is doing in my lab. She has a mouse that basically is a knockout first. It's a mouse that does not make the DNMT3A protein from one of its two pieces of DNA. And then she can turn the gene back on at any time in the development of the mouse and ask, does the mouse get better? This is basically modeling what perfect gene therapy would look like that we would someday try to achieve, and that the biopharmaceutical industry is trying to move towards. So we're going to be doing these experiments to try to give us this idea of if a genetic therapy is the way to go. We're just at the start of this, and I apologize for the research endeavor that it is not fast, but we will be giving you updates as quickly as we can do it. In the last few minutes, I just wanted to briefly touch on the complementary research we're doing, really in collaboration with the TBRS community, because it all starts with biospecimen donation on your part. And this is working with Tim Lay and Kristen Kroll at the Washington University, where I am. Chris Kroll is at the Intellectual and Developmental Disabilities Research Center. And Tim, a number of years ago now, worked with the TBRS community to collect blood samples and create induced pluripotent stem cells, as Dr. McGraw was talking about. And we've been starting experiments to study how TBRS-derived induced pluripotent stem cells are affected when you start making neurons from them. And we do this for three big reasons. One, mice are not humans, and we may make some discoveries in mice and understand molecular mechanisms, or think we understand molecular mechanisms, but they can be different in human cells. So we can immediately sort of check our math from the mice and anything that's a cellular assay in the human cells and make sure that it holds true at a cellular level in human cells, so that we don't go down a path with the mice that is not fruitful. We can also study some things that the mice aren't really showing that makes them not a good model. One thing I didn't mention is that the mice do not have an enlarged head or an enlarged brain, even though in TBRS that is a sort of key clinical feature. So we want to look at these cells and ask, when we make them into neurons, do they overgrow as you would expect in an individual with TBRS? Maybe these human cells have a bigger effect. I apologize, I have a fly attacking me. So I've been trying to not acknowledge it. But we can study the growth of these human cells, and the difference in brain growth between mouse and humans is known to be very big. That's why we have big brains, right? Mice do not have this, sort of relatively speaking. And so this may be one place where the human cells really help. And then finally, once you have good phenotypes, sort of in-a-dish phenotypes, as Dr. McGraw was talking about, you can test the therapeutic interventions that we're hoping to generate. So very quickly, we've sort of worked with the pipeline, starting with the TBRS community, making the donations, and then Tim Ley really generously using existing infrastructure he had to study AML and DNMT3A and generate these cell lines, and then handing them off to Kristen Kroll and I to study them in neural lineages, to make neurons and other neural cell types. And really this is driven by Chris. I tend to be the intermediary that recruits people to work on the work and then continues to be a cheerleader, so I get to report on it, that we're making success, having some success. So we've generated 2 different lines that actually match the mice that we had made in my lab, or that Tim Ley made, and we collaborated together to study, the 882 and the 904. And then we've made controls for these lines. So we corrected the missense variant so that we have cells that are genetically identical except for the mutation. The mutation that was associated with TBRS is gone. This is sort of a gold standard control for studies of these cell lines so that we can know exactly what was driven by the variant. So we have these 2 lines, and we've been studying them, and Gareth Chapman, who's a really talented postdoc in Kristen Kroll's lab, has started making different types of cells, as you were hearing about from Dr. McGraw. We can direct them to become inhibitory-like neurons and go through a developmental process like becoming an inhibitory neuron or an excitatory neuron. And then we look at their growth, and we start to look at their function and their molecular changes. It's very early days. We're actually sort of in the middle of fully validating the lines, making sure they're everything that they should be at a genetic level. At the same time, we're starting to do functional assays. So, take this with a grain of salt that this is a work in progress. But we are seeing some interesting effects. For example, particularly in these, when you're making and developing inhibitory neurons, seeing overgrowth of these neurons, you're creating too many at the neural progenitor cell stage, which would be the equivalent of the sort of early developing brain. So we'll be able to study these effects and understand the underlying molecular changes, and I spoke a little bit about that yesterday. If you're interested in a deeper dive, you can go back and watch that in the recorded material. So I'll end there, because I'm over. I do that a lot. Just want to say thank you to the lead authors who are really driving the work. Diana, who's been sort of a remarkable engine for TBRS research in my lab, and then Kim, who's picking up the mantle with the research. The other folks at WashU that we're working with, the entire Gabel lab, and a special thank you to Tim Ley, who's a remarkable collaborator and has been sort of helped us get going with the IPS work, and Chris Kroll, who has this expertise that we can access at WashU and helps us do the experiments the right way. And finally, TBRS community, really our collaborators that make all that possible and really provide an inspiration that makes it just the next level science that hopefully we can sort of continue on providing insights and moving towards the idea of therapeutics in the future. So thank you very much. Happy to take any questions.